We're here with the National Library of Medicine, and we're going to read Synthetic Living Machines, A New Window on Life. Increased control of biological growth and form is an essential gateway to transformative medical advances, repairing of birth defects, restoring lost or damaged organs, normalizing tumors, all depend on understanding how cells cooperate to make specific, functional large-scale structures. Despite advances in molecular genetics, significant gaps remain in our understanding of the mesoscale rules of morphogenesis. Now, most people don't know what morphogenesis is, so let's look it up. We're going to Google morphogenesis and click on the Wikipedia link. Morphogenesis, from the Greek morpho shape and genesis creation, literally the generation of form, is the biological process that causes a cell, tissue, or organism to develop its shape. Now, back to the article. An engineering approach to this problem is the creation of novel synthetic living forms, greatly extending available model systems beyond evolved plant and animal lineages. Here, we review recent advances in the emerging field of synthetic morphogenesis, the bioengineering of novel multicellular living bodies. Emphasizing emergent self-organization, tissue-level guided self-assembly, and active functionality. This work is the essential next generation of synthetic biology. Aside from useful living machines for specific functions, the rational design and analysis of new coherent anatomies will greatly increase our understanding of the foundational questions in evolutionary development and cell biology. The subject areas here are development biology, bioengineering, and synthetic biology. Now, let's scroll down to this image. Synthetic functional morphologies. You have biology, computer science, synthetic morphology, cybernetics, cognitive science, and their tools are optogenetics, organoid culture, nanomaterials, and computational modeling, and in silico evolution. And what impacts does it have on drug discovery and disease modeling, decoding design principles of human development, evolution of body plans, regenerative medicine of birth defects, injury, and cancer normalization, new AI platforms and algorithms, swarm robotics, and useful biobot functions. Now, to figure out how we can create the morphogenesis we want, instead of the morphogenesis your cells would do on their own, we're going to come over here to National Library of Medicine, and we're going to read Cracking the Bioelectric Code. Patterns of resting potential in non-excitable cells of living tissue are now known to be instructive signals for pattern formation during embryogenesis, regeneration, and cancer suppression. They are playing with metamaterials. They are changing things back and forth, like in the days of Egypt. The development of molecular level techniques for tracking ion flows and functionally manipulating the activity of ion channels and pumps has begun to reveal the mechanisms by which voltage gradients regulate cell behaviors and the assembly of complex large-scale structures. A recent paper demonstrated that a specific voltage range is necessary for demarcation of the eye fields in the frog embryo. Remarkably, artificially setting other somatic cells to the eye-specific voltage range resulted in formation of eyes in aberrant locations, including tissues that are not in the normal anterior ectoderm lineage. Eyes could be formed in the gut, on the tail, or in the lateral plate, mesoderm. These data challenge the existing models of eye fate restriction and tissue competence maps, and suggest the presence of a bioelectric code. a mapping of physiological properties to an anatomical outcome. This addendum summarizes the current state of knowledge in development bioelectricity, proposes three possible interpretations of the bioelectric code that functionally maps physiological states to anatomical outcomes, and highlights the biggest open question in this field. We also suggest a speculative hypothesis at the intersection of cognitive science and development biology, that bioelectrical signaling among non-excitable cells coupled by gap junctions simulates neural network-like dynamics and underlies the information processing functions required by complex pattern formations in vivo. In vivo means in real time. Understanding and learning to control the information stored in physiological networks will have transformative implications for developmental biology, regenerative medicine, and synthetic bioengineering. It has long been known that all cells, not just excitable nerve and muscle, drive and respond to slow changes in transmembrane potential. Ion channels and pumps segregates charges to opposite sides of plasma and organelle membranes, producing slow changing differences and resting potential among cells in vivo. Indeed, spatial gradients of transmembrane potential distribution exists also at the level of tissues and organs, and have been long known to correlate with important events in pattern formation such as gastrulation, neurogenesis, and limb induction. The classical data on development roles of endogenous bioelectric signals have recently been revitalized by the development of molecular resolution genetic and pharmacological tools for the investigation and functional control of ionic signals in vivo. Changes in resting potential regulate differentiation, proliferation, migration, and orientation of a wide variety of cell types, including stem cells, neurons, and neural precursors, and migratory populations such as neural crest. Recent data have implicated spatiotemporal patterns of transmembrane potential in the regulation of embryonic development, regeneration, and metastatic transformation. Thus, bioelectric cues function alongside chemical gradients, transcriptional networks, and haptic/tensile cues as part of the morphogenetic field that orchestrates individual cell behavior into large-scale anatomical pattern formation. This is what regenerative medicine is. This is life extension technology.